Yeah, I think this might be officially the least amount of time I've been given to prepare for a talk, but I would like to thank Philip specifically for this opportunity, unexpected opportunity. So I'm Addie Thomson. I'm from Michigan State. My program focuses on maize and sorghum primarily, although I'll talk to you about a few other things today. And so I come to you just as a way of introduction here as mostly a geneticist, but our program does a lot of improving approaches for prediction, utilizing, among other things, plant phenomics. What this looks like in a practical sense is that we do a lot of field-based phenotyping for the most part, and produce a lot of our own lines and varieties, do some pre-breeding. This is around 18 U.S. acres per year. So I started at Michigan State back in 2018, and in addition to setting up a field program from scratch that didn't exist before, I also had to set up a team of collaborators. And as you all know, as phenomicists, we need a lot of diverse skill sets in plant phenotyping. So back in around 2019, I put together a group of plant scientists, horticulturists, breeders, molecular biologists, as well as computational math, science, and engineering, statistics, probability, electrical, computer engineering, etc. And we ended up getting an internal grant to work on a project where we were funding not necessarily any sensor development or even really funding the deployment. We were just purchasing some pre-made sensors, putting them onto drone platforms that we had, and then funding data acquisition. Because we knew that we had a lot of breeders who were doing really cool science and had a lot of data sets they were already collecting data for out in the field. And we knew we had a lot of people in the computer sciences who were developing really exciting algorithms, and they were going elsewhere to find large data sets to test their algorithms. So we thought, why not just put some data in the middle of these groups and then let them form these teams. So Michigan State, there's not a lot of us here, you'll notice, but it's because we've started working on these things, but I think until recently, no one has really called it plant phenomics. So I think it was very apt that you had the discussion in the working group earlier this week about plant phenomics as a science, because that's still an ongoing development, I would say. So one of the offshoot teams that I was involved in was with the Remote Sensing and Geospatial Information Systems group. So they fly the drones, but we collaborated with some geneticists, some statisticians, machine learning, deep learning, and in particular, the person in the upper left, Daniel Morris, who's in engineering at Michigan State. And one of the big questions that my group had been thinking about is that from the perspective of plant breeding and genetics, we really want to know how a variety is going to perform in order to assess its usefulness, ideally with as little effort and input as possible, but So there are existing algorithms like genomic prediction where we use the genetics to predict phenotype. But we know that that doesn't really perform well in new environments because you're skipping through all the stuff that happens in between, which makes no sense, really. We can also use physiological models, like crop growth models, to really simulate how a variety is going to grow in its different environments. The issue there is that it's going to rely on a lot of tedious hand-measured phenotypes to parameterize those models. So our question was, can we acquire those phenotypes some other way, those parameters? So if we can acquire the parameters using a high-throughput method, and then parameterize something like a crop growth model to predict those composite traits out into another environment. So again, I started thinking about what would be some approaches that you could use to acquire those types of traits. And so this was my mental map at the time, where I was thinking, okay, there's these different sensor technologies that we have access to. There's different traits that we could measure that would then accumulate into these modeled traits and finally into these composite traits that are less heritable and more influenced by the environment. And the reason we were thinking about it this way, I'd had experience with a few of these different sensors before, but the one that was really missing in my mind was this LiDAR piece. I had guessed it could do all these different things, but I had no evidence of that. But the reason we suspected this was based on some work at Purdue a number of years ago where we were looking at sorghum and looking at different varieties, and we pulled out of the RGB orthomosaics. So you structure from motion, you've got the overlapping images, so your pixels are estimated in the Z direction as well as the X and Y. So from those pixel level heights across the plot, we built histograms of what the height was doing across the plot and came to realize, as was shown nicely in a talk the other day, that you get a lot of other information out of those histograms. Sometimes you have a leaf at the top that's very flat. Sometimes you have really skinny panicles or broad panicles. Sometimes you have tillers. And so we were able to see a lot of that in these histograms, including down to when we had something like an escaped dwarf in a field. So this is actually a picture from the plot where that histogram came from. So we thought perhaps it's possible to use information about distributions of pixel levels in the canopy to learn more about what's going on in the canopy. So that was the question and the test. To do this, we knew we wanted to parameterize on a really, really specific level. And so we actually took a diversity panel. This is in corn now instead of in maize, instead of in sorghum. And we actually marked plants in the field in thousands of plots, and we counted leaves and measured leaf areas. The reason we were going into the phenology so extensively was because we knew that was an important parameter for a lot of crop growth models. What you're seeing here is in the chart is actually a power function built on just 3 parameters. The blue dots are what we measured in the field, and then the red line is the fit from the power function. So it works very well. So this was back in 2020, we had a number of flights, 7 flights across the season. From this, Daniel Morris actually was the one that generated the voxel model that was looking at these traits through time and then estimating what all of the measured traits that we took could we really accurately predict or estimate. And so in this case, I'm going to cut to the chase because I have other things to talk about. So I'll tell you, it worked better than I thought it would. We were able to predict things like flowering time, ear height and plant height, obviously, ear leaf number, largest leaf length, plant, flowering time, soaking, I said that already. Okay, tassel height, total leaf number. So things that didn't work as well were stand count. We didn't have necessarily, I think, the appropriate measures for that, and then the largest leaf width. But what you're seeing here actually is a comparison between if we were using genetics to predict that same information versus using the LiDAR to predict it. So the blue bars are the LiDAR. And in a lot of cases, we're really outperforming the ability of what the genetics can do, which is exciting. Although it's not exactly a fair comparison, which I'd be happy to talk about later. But the overall thought process, and I'll warn you, this is an unfinished story. So this is really a call to action to anyone else that's interested in process-based models, that I think this is an interesting paradigm to think about as you're thinking about what traits you're going to measure and how you might integrate those into a breeding pipeline, for example. So in our case, what I would really love is if we were collecting phenotypes that are informed and targeted, not necessarily the kitchen sink for some cases, to train the genomic prediction models on those key traits that don't respond as much to the environment, and then to parameterize the process-based models for each genotype using those parameters. So that could allow you to include actually predicted and simulated genotypes. It wouldn't have to be ones that you already have observed. And then using the process-based model to integrate the soil, climate, and management data along with those component traits to predict things like yield that aren't as easy to predict using genetics directly. And that allows you to then transfer those predictive abilities to other locations, soils, climates around the world and even into the future. This is not necessarily a new idea. The companies are doing this, I can tell you. But I think it's an area where both phenomicists and plant breeders and plant scientists, as well as physiologists, can all come together to make meaningful progress. So I have a postdoc right now who's been applying this in sorghum as well, and she's done some interesting work in also measuring photosynthesis and stomatal conductance and relating that back to hyperspectral imaging. I'll warn you, I'm not going to go into a lot of detail on a lot of projects, mostly because I don't want to bore people, right? I don't want to tell you, here's the systems, here's the details of the projects. Excuse me. So I want to give you just some tastes of ideas of ways that we're implementing some areas of interest, right? So another area is when I first came to Michigan State, there was a new disease called tar spot. This is caused by a fungal pathogen. It's been around in Central and South America for the last 100 years, but it was new as of 2015 to the upper Midwest United States. And so in addition to doing some pre-breeding and genetic screening, we also were working to collect data throughout the season and then relate that back to onset and severity, with the idea that maybe if we could identify disease before it's visible in the field, you'd have more management options. As those of you who have done work like this before will probably know, it's challenging because you can do a really good job of saying these plants are stressed, but less of a good job of saying specifically what it is that's stressing the plant. It does enable for a large farming operation to point to places to scout, which is important. But where we've been moving lately, though, is more in the line of can we, for breeders, easily quantify that trait of severity so that we have a numeric trait for people to work with for breeding. But it is a pretty impactful disease. This is an example from 2018 where you're seeing a field and that brown area is heavily infested, and then just a couple weeks later, before it should be turning brown, the whole field is senesced and that area has lodged, fallen over. Where we have found a lot more success with spectral imaging and modeling is in this leaf level hyperspectral imaging for nutrient content. We have a lot of interest in the lab right now in looking at nitrogen response in maize, and so we've been looking at how do the leaves build up nitrogen throughout the season and then remobilize it toward the end of the season, and do they keep uptaking? Do they stay green? So one of the ways we do this is with these leaf-level hyperspectral scans, but I will mention that right now, as fairly naive geneticists, we're mostly using these beautiful scans as average values across the leaf. So I think there's a good area of exploration to be had for people who really want to build image-based modules that would take advantage of the texture and the distribution of those pixels across the leaf, because we know that leaves stripe and have veins and things in different ways based on their biochemical properties as well as their physical properties. And so I think there's a lot of room for improvement in models like this. It looks like I have a really excellent model, but we're modeling water content here, so it's not that hard, right? Water, nitrogen, we got those down, but there's a lot of other opportunities. So this is at the field scale what this would look like. We're looking at different hybrids and inbreds and doing screening and breeding and development towards really low input nitrogen maize. And so that's where one of the current interests is in the lab. Other research projects going on in my lab and at Michigan State, just quite briefly here. We just had a few days ago a multi-objective genomic mating optimization software pipeline come out. So if you're interested in that, it's called PiperOps, just a little plug. We're looking at nitrogen and biostimulant in combination, and we have a big project right now on flavonoid content in maize and sorghum. I wanted to highlight just a few other projects that we're doing in collaboration with other groups, both to advertise some other folks from MSU, but also to get you thinking about other possibilities. So one project we've been doing the last few years is with teff. Ergostis teff, this is native to Africa, and it's important for making, in Ethiopia, for making injera, which is a spongy bread that's used in a lot of the meals. And what Bob Van Buren did is he put together a diversity panel of different accessions and worked to do some screening and phenotyping, high throughput phenotyping, as well as the sequencing to build this up as a population for a resource for people to use. And one of the things that we've been interested in is not only nutrient content, but nutrient availability, as well as things like things that you think should be simple, right? Like brown versus white seed, which turn out to be not quite as simple as you might guess because the phenolics in those don't match what we anticipated. If you know a lot about phenolics and teff, I'd be happy to talk with you. Another fun example is from potato. This is with Eva Faré. She has a talented postdoc right now working on this. They actually imaged Arabidopsis plants frequently over time to look at diurnal cycles. And these are a combination of domesticated and wild potato plants. So they're baby potatoes. They're not Arabidopsis. They're baby potatoes. And what they did was they looked at them under different light cycles, but they were watching the pixels of the plants because as plants wake up and fall asleep, their leaves kind of do this, right? And so they're able to plot over time the changes in the pixel areas. And what you see is it's going to go like this, right? Because the plant's going to grow. So if you take that function out, you're left with this diurnal cycle. And so they've been phenotyping clones of these populations of potatoes, which we also have sequence information for. And so we're working on doing GWAS on those potato varieties to look at what genes are influencing circadian rhythms. So as I was thinking about phenomics as a field, as a science, one of the things that I think of, and I know it's not the only thing involved in phenomics, is addressing bottlenecks. And I've shown this slide a few times, but I really like the concept of thinking about things in terms of a bottleneck, because it forces you to really address what is the problem and how are you going to solve it. So the problem for me oftentimes is going to boil down to either people and skills or equipment and tools. And if it's people and skills, I ask myself next, okay, does this expertise exist? If it does, make a collaborator. Find someone that knows how to do it and learn from them. Work with them. That's a great way to do it. If it doesn't, then you're going to have to train and study, right? Or if you don't know, you need to ask around. This seems really straightforward, but you'd be surprised at the number of times, especially students possibly, but a lot of faculty as well, don't sit through and think through these steps. For equipment and tools, does this tool already exist? Is there a really good solution already out there? If there is, just try to buy it, right? Don't try and reinvent the wheel, buy the thing. If it doesn't, then you can build it. And if you're not sure, test some things. Now this is gonna interact, obviously, because if you're not able to build something, you're gonna have to collaborate. But what I like to focus on here is that oftentimes students in particular focus on this central part where you assume that you need to do the training and the studying, build the knowledge and the skills, learn critical thinking, which is super important. And that is absolutely true. But it can't be understated that in science, some really important pieces of this are communication and confidence and learning creativity. Those aspects, I think, are critical. And then, as always, you're going to have to weigh time and cost. So I wanted to present a few choice quotes and perspectives from some people that I sort of admire. And in thinking about this communication, confidence, creativity area, a person that came to mind is Temple Grandin. I'm not sure if you know who Temple Grandin is, but she is an autistic scientist who's an animal scientist who innovated on ways to work with cattle processing facilities. And one of the things that she says is that the world needs different kinds of minds to work together. When different minds work together effectively, there can be great successes. They complement each other's skills. And what this really boils down to for us is that a lot of times you need to have extra time to find a common language with your collaborators. You don't know each other's expertise. You have to build that over time. Another couple of people I'm going to highlight real quick, Dr. Ron Phillips. Always be above reproach. Conduct your research in a way that you trust your data. And if you see something weird, you can take advantage of that instead of assuming that it was an error. And also care about the person, not just the scientist. And finally, Dr. Norm Borlaug, who focused on training, education, and youth. And I think it was an interesting perspective from him that soft skills are essential and can come from experiences outside your formal training. He was really big into youth baseball and wrestling. So actually he was the one that I think brought wrestling to Mexico, I think. But he was big into these youth sports and building teams and getting people to learn in that sense. So I guess my take home for this topic here is that you're here at this conference. Maybe it's your first conference, maybe it's your 201st conference, but network, have fun, build a community of peers and mentors, and help combat imposter syndrome in yourself and others. That's a big one. The way that we do that in my lab is that we have a lot of outreach programs we participate in. One of these is Ecotech Labs out of Detroit, which brings high school students into our lab to work. We also participate in this multicultural apprenticeship program, and we work with a number of students and farmers and teachers. One last thing, real quick. I wanted to highlight one of the classes that I teach where we work with data sets. We bring together computational and plant scientists to do things they've never done before and that have never been done before. Here's an example using LiDAR data and hyperspectral imaging in popcorn and potatoes, respectively. But I highlight these because the students came to me with things that looked crazy, and they said, what's going on? Where's the corn? And it turned out they had all the infinite returns that they hadn't filtered out of the LiDAR. Or in this example, we had a lot of soil pixels, obviously. I said, those are not plants. So we were able to find out where the problems were. I translated this into an undergrad course last year, and so in February I had students with me that were at the North American Plant Phenotyping Network meeting. Okay, with that, I will end. Thank my funders, and thank you for listening if you have any questions.
So thank you very much for the talk, and where the frontiers are. Are there any questions? Here in the middle.
Addie, thank you very much for your nice presentation. I agree a lot with your idea of trying to integrate high-throughput phenotyping observations into models. Sometimes the kind of traits that we estimate for high-throughput phenotyping are not exactly the kind of variables that are simulated by the model, so that there is a gap between them. How far do you think that they are currently, the kind of traits that we can access from high-throughput phenotyping, from the kind of models that you are trying to use in mice? How do you think that we could fill this gap?
That's a great question. I think a lot of it is within some amount of reach. We've been working on trying to figure out what we're missing and how we can get it. And we went into a lot of detail trying to measure really specific leaf angles and modeling the arches and everything. And then looking at photosynthesis, trying to get at resource use efficiency. Nothing has quite closed it yet. For us, we're still missing part of the people component. None of the people in my lab currently are really crop growth modelers. So if there's a crop growth model in the room that wants to collaborate, I'd be happy to talk to you. But it's a good question. I don't think I know the answer. What is the gap? I don't know, but we're working on it. For me, I struggle between what is the best that can be done and what is good enough for a breeding program. And, NGVI gets you really far, which is frustrating because I want the answer to be cooler than that.
Yeah, that was a great talk. A lot of cool ideas and some triggers. I liked your scheme with I made a picture, 1 second, the what is the problem slide, right, you addressed. I think there is something like if the tool exists, people are not aware of it, but there is a reason maybe that they are not searching for it. It is maybe not listed, but I think it is fun to build your tool yourself. Right? That's a big reason, I think. And it's also the not invented here syndrome. That is maybe even more of an influence than your imposter syndrome that you mentioned on the latest slide. And that is very difficult to overcome.
What did you say? The what invented here? What?
The not invented here syndrome.
Oh, did not invented here. Okay.
What do you think?
What do I think? I agree that a lot of what we all probably got into this field because we thought it was fun, right? If you didn't think it was fun, you wouldn't still be here. So I'm not saying don't do the stuff you find fun, right? But at some point, you're going to have to articulate to either your boss or a grant panel why it is you're doing what you're doing. So you have to be able to measure the fun, balance the fun with the practicality. And so I do think there's room for new tools and innovation, even if there's something that already exists. You just have to articulate why, right? Because I think if it's only for fun, that's going to be a challenge to get funding and get people excited about it. Other than you, I mean, I'd be excited.
But on the other hand, if you're a PhD student, then you have plenty of hours and you don't have budget. You could always spend more time than money on your problem.
PhD students, the pay is going up, so the time is valuable. And at least my students spend quite a bit of time in the summers in the field and quite a bit of time in the school year in classes. And so really, time is always going to be your most valuable resource. Maybe that's different at other places, but at least here, that's really, yeah.
Yeah, cool. Thanks. Yep.
Thanks.
Then let's thank again for the nice talk. Yeah, it's super. That's it.